Hello friends, today we are going to start molecular biology, and in this video lecture we are going to discuss about DNA. So without any delay, let's start our today's topic. So firstly, where DNA is present. So DNA is present inside the cell. So if I talk about that, if it is a cell, so inside the cell there is a nucleus, and inside the nucleus DNA is present in condensed form. So this is a chromatin fibers which is present inside the nucleus, and inside the nucleus we can see this chromatin fiber which is made up of DNA and histone protein. So DNA is negatively charged and the histone protein is positively charged. That's why DNA wrapped around the histone protein, as they both are having opposite charges, so they both are attract each other and wrap around each other and form a chromosome like structure. So when the chromatin fiber is condensed in the most condensed form, it forms the chromosome structure. As you can see, this is a chromosome structure. If we cut this part, or if we if we open this part, so we can see this like this DNA structure. So DNA is just condensed and fully condensed and form the chromosomes, and we can, if we see this small part of chromosome, we can see like this DNA. So DNA is made up of different bases. So four bases is present in the DNA, and phosphate linkage is also present, diphosphate linkage. So four bases like purines and pyrimidines are present inside this. So G means guanine, make 3 hydrogen bond with cytosine, and adenine make 2 hydrogen bond with thymine. So like this, it's a basis of the DNA's form. So now let's talk about the condensation of DNA. So condensation of DNA, if we see, this is a DNA whose width is 2 nanometer. It's a short region of DNA. We can see this double helical form of DNA. So DNA is also known as the unit of inheritance, and it is responsible for the inheritance of characteristics from parents to offspring. It carries the characteristics. So it is, that's why it is called as unit of inheritance. So it's, after this, when it condenses, more condense and form the bead on string form of chromatin. As we can see, this is a histone protein over which, around which DNA is wrapped like this, and just like the beads, histones just like a beads, and the string is just like DNA is just like a string which is wrapped around the histone protein. So beads on string structure it forms, and the width of this is 11 nanometer, and this model is called known as nucleosome model. After this, when it condenses, more condenses and form the structure like this, this is a histone, and over which uh, DNA is wrapped and form like this structure. This width is 30 nanometer, and we call it as solenoid model. So 30 nanometer chromatin fiber of packed nucleosome, this is known as solenoid model. After this, section of chromosome in an extended form, we can see this form when form the sections like this of chromosome in the extended form. It is known as scaffold structure or scaffold model, we can say, and its width is 300 nanometer. So when it's all, then more condenses and form the structure like chromosome, condensed section of chromosome like this. We can it is having 700 nanometer width. So after this, we are having the entire mitotic chromosome like this is structure of chromosome, and in the middle, in the center of this, centromere is present, and its width is 1400 nanometer. So it's the fully condensed form of DNA. So this is all about the condensation of DNA, which we have talked, which we have discussed, like this DNA 2 nanometer, after this histone nucleosome model which is 11 nanometer, and then the solenoid model 30 nanometer, the scaffold model 300 nanometer, then the full condensed form of the chromosome section like 700. So this is of 700 nanometer. Then the entire chromosome, we can see this, and it is a 1400 nanometer width. So this you can see like zigzag pattern, or we can say the helical form. This is the DNA which is wrapped around, and in condensed form it forms a chromosome like structure when it condenses, and inside and between this, in the center, centromere is present, which keep the both chromatin together. So after this, we discuss about overview with respect to the existence of gene. So firstly, in 1865, unit of inheritance, which is discrete unit of inheritance, was discovered. In 1880s, chromosome was discovered by Sutton's scientist. In 1903, homologous chromosome was discovered, and in 1909 to 1911, crossing over was discovered. As we see the crossing over in during meiosis prophase, during pectin, crossing over, which is responsible for the exchange of gene from the chromosome. So this was discovered in this year. In 1911 to 1913, there was a discovery of gene that we can map gene in order to, along the chromosome length of chromosome. For example, if this is a chromosome, so along the length of the chromosome we can map that from there to here, this gene DNA is responsible for this character or for that character, for any character which can be mapped. So around the along the length of the chromosome, we can map the gene, that which gene is responsible for what character, so we can map that. In 1944 to 1952, we discovered that DNA is a genetic material, and in 1953, the discovery of was DNA structure was discovered. From X-ray crystallography method, we discovered this DNA structure. So now come discuss about the chromosome as a carrier of genetic information. So measures from pus cells, he isolated nucleic acid from pus cell. Then the Griffith experiment, which was also known as transforming experiment or transforming principle, where he proved that Griffith experiment proved that the bacteria having rough surface can't able to infect other organism, but this bacteria having the smooth surface, slimy mold surface, slime like surface cell wall, which is responsible for the if infection. So from this his experiment, we come to know about the this is something is responsible for the infection, which from an infected to an infected person, which goes from infected to an infected person and can cause disease. So after this Hershey and Chase experiment, which fully completely proved that DNA is a genetic material, where he uses bacteriophage, and he proved that DNA is a genetic material. So thank you, that's all for today. Thank you for watching.